I'm going to be talking today not about cancer, but I'm going to be talking about coronaviruses. So just very quickly, I kind of like want to state like what we do in my lab. So our focus is on applying combinatorial optimization techniques to answer questions and solve problems in biology. So this is like super broad. We mostly specialize on evolution, and in particular cancer evolution. But a key theme is we like, clean, beautiful computer science problems. And the challenge is actually, how do you go from a biological question, how do you translate that into a computer science problem? And oftentimes, you're really lucky and you get a beautiful computer science problem that is kind of like new and needs to be studied. And at the same time, that computer science problem leads to, really cool biology. So I'm going to be talking today about recent work that we published at Recomp last year, and the extended journal version came out in MBE. So this work was led by two graduate students, Troy and Palash, and we also had help from a bunch of undergrads. So let's talk about it. So we're looking at coronaviruses here. So this is a phylogenetic tree of different coronaviruses. So the taxonomic order of coronaviruses, they're called, it's called Nidoviralis. So nido comes from Latin, nidus, and means nested. And this is actually related to how coronavirus genomes are structured. So by the way, when I say coronavirus genomes, like SARS-CoV-2 is a coronavirus, but there's many other coronaviruses. So what I'm going to be talking about is tools that apply to the coronavirus family. So that's beyond SARS-CoV-2. So let's take a look at coronaviruses. So coronaviruses are RNA viruses. So it's an RNA genome. The genome is about 30 kb long, and it's a positive sense RNA molecule. So this means that it's ready to be translated by the host ribosome. So as you know, one messenger RNA leads to one protein. So when the ribosome starts translating this coronavirus genome, it will start from the 5' end, from the left. And it will keep on reading, and one of two things will happen. So the first start codon it will bind to and it will start translating is here. It's at the beginning of ORF1A. And there's a stop codon at the end of 1A. But in front of that, there's a slippery sequence. So it causes like a -1 frame shift with a certain probability. So you get one of two products. You get either 1A, or when the frame shift happens, you get 1AB. So if you look at this, this is just only one part of the genome. So this is actually called the non-structural part. Like downstream in the genome, you actually see other genes, and some of you will recognize S, the spike protein. So that's not being expressed initially. So that leads to a big question, like how are these downstream structural and accessory genes expressed? So what's actually happening is that in 1A and 1AB, this is a long polypeptide, and when it falls into space, it cuts itself up into little pieces. So you get about from 1AB, there are about 14 to 15 non-structural proteins, and 3 of those non-structural proteins form a complex called RNA-dependent RNA polymerase, RDRP. So what RDRP does is super interesting. So it takes as input RNA and makes a copy of RNA. So if you remember from like your biology classes, the central dogma of molecular biology, DNA goes to RNA and RNA goes to protein. So there is there's nothing in eukaryotic cells that basically makes copies of RNA. So that's what this virus provides. So that's part of every, so actually this RDRP, it's a feature of RNA viruses. So what happens with RDRP is super interesting. So, it takes RNA's input and makes a copy of RNA. Now let's zoom in a little bit. So suppose it's actually making a copy and it starts at the 3' end, and it starts reading from right to left. And I want to zoom in. Suppose it hit this point over here. So we're looking at just in front of spike. So there's this red box over here that I call a transcription regulatory sequence. And you'll see that the same sequence occurs in different places in the genome. And what actually happens is super interesting. So at this sequence, so this is the template strand. So it's making a copy of the template strand, and it's producing a new strand, the nascent strand. And you can kind of like see complementary base pairing happening. So at this point, what can happen is something known as template switching. So rather than continuing on, to the left, like moving on to 1B and then 1A and the rest of the genome, it can actually undergo template switching. So it can actually make a jump to the beginning of the viral genome. So this 5' end over here. And when this happens, you get a transcript that looks like this. So it has the leader part, and then it skips over 1AB. So when the ribosome translates this subgenomic messenger RNA, it will produce the spike protein. And this happens, upstream of every accessory and structural gene. You have this TRS sequence. So I'm showing you here the TRS sequences and multiple sequence alignment of them. And the key point is, in coronaviruses, we have discontinuous transcription due to template switching of RDRP at transcription regulatory sequences. So, this led to two questions that I'm going to talk about in today's talk. So question number one is, if we're given a coronavirus genome and we know the gene locations, can we identify these transcription regulatory sequences? And the second more interesting question is, what if we're given, an unannotated genome? We're just given a genome sequence. We don't know what the genes are. Can we simultaneously identify genes and transcription regulatory sequences? So these are the two questions that we're going to be focusing on today. So question two is especially relevant if, if you identify a new coronavirus species. So you've never seen it before, you just assembled a new genome and you don't know what's going on. So by answering question two, you can actually understand, what are the different genes that are present and, what are the different transcription regulatory sequences. So you can gain more understanding of the of your new viral sequence. So, before our work, there were basically, so essentially the two questions that I'm asking here, they correspond to two different, well-studied topics. So the first question about finding the transcription regulatory sequences is essentially like a motif finding problem. So we want to find a subsequence that occurs in a genome. So there exist many methods for doing this. So meme is a very well-known general purpose method. And in the space of coronaviruses, there was a paper, Super, in 2021, that was specifically designed for finding motifs. But it uses a lot of prior knowledge. So it actually, you need to specify what species or what genus of coronavirus are you looking at. And you also need to give it secondary structure. So that's the space of motif finding. And in terms of question two, finding genes, there's a lot of gene finding methods. Protocol, Glimmer, and more recently VADR, which is specifically designed for viral sequences. So, we saw a gap here. So there was no method that combines motif plus gene finding for coronaviruses. And our hypothesis was that by, so by looking at TRS sites, by using that as an additional source of signal, we should be able to, find genes more accurately. So that's an additional source of information that can be used. So let's talk about that. So I'm going to start with the first question, which is finding just the transcription regulatory sequences, and then we'll move on to the second problem, which is about finding the transcription regulatory sequences plus genes. So let's talk about this first problem. So the key object that we're looking for is something that we call a TRS alignment. So a TRS alignment is a constrained multiple sequence alignment, and it differs from a regular multiple sequence alignment. So the differences are as follows. So first of all, the first sequence corresponds to the leader. And we don't want to have any gaps in the leader sequence. And the reason why is because we need, we basically, there's no wiggle room here. So when template switching happens, we basically have bases that pair up with one another. There might be mismatches, but there's no room for gaps. If there was a gap, an insertion or deletion in either sequence, we would be in trouble. It wouldn't fit perfectly anymore. And the same constraint also leads to, the same biological constraint leads to number 2 over here. Like for the internals, for the body sequences, so A1 to AN, we cannot have internal gaps, only flanking gaps. So you can span different parts of the leader, but you cannot have internal gaps. And moreover, if you look at your alignment, there is something called the core sequence. So the core sequence corresponds to the largest subsequence in A0 that is not paired up with any gap in the remaining sequences. So the core sequence here begins at is AACG So the length of it is 7. You cannot extend it. If you would extend it to the right, you would get a gap, and vice versa for and similarly for the left region. So the way we score a TRS alignment is different from a regular multiple sequence alignment. So typically, multiple sequence alignments are scored using sum of pairs. So you would look at all pairs of sequences. In our case, because we have template switching with respect to the leader, we only score with respect to the leader. So we look at matches and mismatches between body sequences and the leader sequence. So for instance, if you use a scoring function that assigns a score of +1 for matches and a penalty of -2 for mismatches, these are the scores you would get. So the score of this alignment is, for instance, 79 over here.
Hi, Mohammed. Can I
Yes.
talk to the...
Yes, please.
So does this, just for what you're talking about right now, it assumes you've identified the transcription regulatory sequences, and now you want to build an alignment. So you've carved out the other pieces of the sequence so that you know they'll be flanked by gaps?
Yes. So I'm kind of like jumping right into the middle. So what I'm doing here is I'm kind of like building a problem statement. So the object that we care about is this TRS alignment. We don't know how to find it yet, but we know how to score it. So like the natural thing, what we'll do next is, find the TRS alignment with a maximum score.
Okay, and then could you just remind what's the L and B difference? Thank you. The TRS-L.
Yes, thank you. Thank you for the question. So the difference between L and B, so there's a difference between, the first TRS sequence, so that's A0, and the first one is at the beginning of the genome. So we don't know its location. So here I'm assuming, suppose we actually knew its location. But the first one is called the leader. And the TRS-B that I have here, B stands for body. So the leader is the beginning of the genome, and the body is the rest of the genome. So that's the difference between TRS-L and TRS-B. And the way we score it, it's TRS-L versus every other TRS-B. So that's what you see over here. That's the scoring function. So that's the big difference. So if you compare a TRS alignment to a multiple sequence alignment, there's constraints on the presence of gaps. So no gaps in the leader sequence in A0, and no internal gaps in the body sequences. And also the scoring is different. So we score with respect to A0. So it's kind of like a star alignment. You could think about it that way. But let's talk about the problem. So we don't know what A0 is, and we also don't know what the other TRS-Bs are. So because all our input actually corresponds to candidate regions, which are upstream of every gene. So I'm indicating them here with W0 to WN. And the other thing that we're given is we're also given the length of the core sequence. So we have a constraint on the length of the core sequence. And the task is to find a TRS alignment such that each sequence in the alignment is a subsequence of the corresponding in the corresponding candidate region. And the core sequence has length at least omega. And the alignment has maximum score. That's the problem we want to solve. So this is a multiple sequence alignment problem. And there's something interesting happening. So usually, multiple sequence alignment is NP hard. But in this case, the problem is actually easy. You can solve it in polynomial time. So let's talk about that. So there's a couple of observations. So the first, observation that we have is, a TRS alignment A, must have an induced core sequence CA with length at least omega. So what this actually means is that the input sequences, W1 to Wn, or W0, W0 to Wn, they depend on one another, and we cannot consider them in isolation. However, you can kind of like break this dependency by kind of like using a subsequence U, by sliding a subsequence U through W0, and you require that the core sequence of whatever alignment you find contains U. And the reason why this works is because your final solution actually corresponds, like the core sequence of your final solution is a subsequence in W0 that is spanned, by every other sequence. So your final solution actually corresponds to a specific window. So the solution to TRS ID corresponds to the window U star that induces a TRS alignment with maximum score. So this leads to the following problem. So the constrained TRS identification problem, where in addition, we're also given this subsequence U. And once you do this, you break the dependency. So you can actually, rather than looking at all sequences simultaneously, so rather than having a multiple sequence alignment problem, it actually becomes a pairwise alignment problem. So it's about aligning W0 to WI, spanning U. And the way we're posing this problem, it's a variant of local alignment. There are three differences. And the differences come from the constraints that I imposed on TRS alignment. So the alignment may not contain gaps, and A0 must contain U. So just a quick intermezzo, just to make sure we're on the same page. So global and local alignment. So these are slides I use in my Introduction to Bioinformatics class. So in global alignment, we want to align all of V to all of W. And in local alignment, we want to align a subsequence. We want to find a subsequence of V and a subsequence of W such that our global alignment of those two subsequences is maximum, has maximum score among all subsequences of V and W. So obviously what we're doing here is kind of like similar to local alignment. But there are a couple of constraints. So we don't want to have any gaps, and we must span U. So you can actually adapt the recurrence, the Smith-Waterman recurrence for local alignment to satisfy these constraints. So that's what we did over here. I'm not going to go into the details, but long story short, you can solve this problem, in polynomial time using dynamic programming, and it's pretty much like a simple adaptation of the Smith-Waterman. So we just get rid of the gap cases, and we make sure that our final solution, spans all of U. So we can solve the constraint problem. So this little slide over here, this little cartoon shows you how we solve it. So for a fixed window U, we can basically solve n pairwise alignment problems, and each pairwise alignment problem will be subject to the constraint that U is spanned. So you can actually combine them by moving the alignment around such that the window U that is spanned in each solution kind of like leads to, so you can kind of like combine all these A knots into a single A knot over here, and you insert flanking gaps. So the reason why this works is because flanking gaps, you're not getting penalized for them. They have a score of 0. So that's pretty much what's going on here. So solving one window takes the length of W0 times L, and L is just the length of all the remaining candidate regions. And the solution that we seek is the solution that has maximum score. So you solve this many different times. You solve this, so you slide a window of length omega through W0. So that takes, time linear in W0. And this leads to a total running time of W0 squared times L. So that's how much time this takes. Good. Any questions about this? Okay, good. So let's talk about the second problem. Or actually, let me just, I'm going to jump to results and then we'll talk about the second problem. So so what I'm showing you here are results from 468 coronavirus genomes that we downloaded from GenBank. And let's focus on CORSID A. So I'm comparing CORSID A to two other methods, super and mean. And here I'm showing you the accuracy of finding TRS-L. So are we actually, the alignment that we identify using our method, CORSID A, does it actually contain the right TRS-L? So, so as input, we basically take the gene locations. And you can see that we, outperform mean, and we match the performance of super, but super is using a lot of additional information. And there is like one case that is actually super interesting. So I'm showing you here a solution, or this is a beta coronavirus. And you can kind of like see the difference between, there was a difference in what super identified. So super identified, this to be the TRS-L sequence, and in particular, this to be the core sequence. And what we did is we identified, the core sequence to be actually over here. So you can kind of like see there's no difference in nucleotides of the core sequence. So AACG, AAC, it's exactly the same here. However, the solution that we identified compared to the super solution, our solution is actually correct. And there's two reasons. So number one is that for beta coronaviruses, it's actually known that the core sequence is located on stem loop 3. And this is stem loop 3. And the other reason is we actually downloaded the Illumina sequence, raw sequences, and we aligned them to the reference genome. And we actually saw a split read. So read that was split into two parts, aligned to actually over here. And it also aligned to the TRS-B in front of N. So you can actually see discontinuous transcription happening here. So the long story, or the key message that I want to make here is that flanking nucleotides around the core sequence actually help you identify the TRS-L more accurately, because when you look at this, you can kind of like see that your flanking nucleotides actually give you more information about the correct location of TRS-L. Because what super identified, like the flanking nucleotides, were very different from the TRS-Bs compared to our solution. Good. So let's go back to the questions that I started with. So to summarize, we spoke about question 1. Can we identify TRS-L and TRS-Bs in annotated genomes? And now I want to talk about the second question. So now we're in this case where we only have the genome sequence. So can we identify genes simultaneously with TRS sequences, given just the genome sequence? So this is a harder question. And so let me just give you a little bit of background. So what you can do is, if you have an unannotated genome, what is a gene? So a gene is actually, it has a start codon, so ATG, and then you stay in frame. So you start looking at codons one at a time. So you do like +3, +3, +3, until you hit a stop codon, right? So there's like 3 possible stop codons. So there's 1 start codon. So what you can do is, given a genome sequence, you can actually enumerate all possible ORFs. So ORFs stand for open reading frames. So these are, candidate genes. So these are intervals where the first, 3 nucleotides are ATG, and the first stop codon that you'll hit is, corresponds to the last 3 nucleotides of that interval. So, what we actually have in coronaviruses is, there's a couple of things. So first of all, this is a very compact genome. So there's not a lot of sequences that don't correspond to genes. And the second thing is, genes don't overlap. So this actually, leads to an independent set problem. So what you could do is you can actually, think about each ORF, a candidate gene, which you can enumerate very easily as a separate, as a node in a graph. And you have an edge between two nodes, between two ORFs, if they overlap. And in addition, in this graph, you can assign the length of the ORF as the node weight. And the solution that you seek is a maximum weight independent set. So you're trying to find a subset of nodes that don't intersect, so there's no edges between them, with maximum total weight. And this is using the fact that the genomes are very compact. So you want to find a set of ORFs that cover as much of the genome as possible that don't pairwise, that don't overlap, that are disjoint, pairwise disjoint. So what we wanted to do is we were wondering, okay, that's great, we can totally do that, but we want to infuse like the signal from TRS, from transcription regulatory sequences. So this is like the third point here. So genes should have similar TRS sequences. So whatever independent set you pick, you better make sure that they also have, that they're preceded, that each gene is preceded by a TRS sequence that is very similar across the genes that you pick. So that's kind of like the high-level idea that we're doing here. So how does this work? So again, I'm gonna, before giving you the problem statement, there's a couple of things that I want to do. So I want to go through a couple of definitions. So each TRS alignment induces a unique gene set. So what does that mean? So if I look at a TRS-B, I can actually, from that TRS-B, I can scan for the first occurrence of ATG. So ATG is the start codon. And, that ATG, corresponds to an open reading frame. So if I look at, for instance, this TRS alignment, I can actually do this for each TRS-B. So TRS-B corresponds to A1 to A9 in this example, and get the first ORF that occurs immediately downstream of that TRS-B. So that's the first thing. So each TRS alignment induces a gene set, okay? And this leads to a really cool problem statement. So what we're trying to do here is we're trying to find, so we're given three things. So we're actually given the leader region, V leader. So we kind of like know where A0 is located. I'll talk about that in a minute. So actually, it's very easy. You can get it from using a couple of biological observations. And we're also given the body region. So we kind of like split this genome into two parts, V leader and V body. And we're also given the length of the core sequence. And the task is to find the TRS alignment such that A0 occurs within V leader, and A sub I corresponds to a subsequence in V body. And the core sequence obtained from this alignment must have length at least omega. And then here's the objective function. So the alignment induces a set gamma A of genes with maximum genome coverage. So genome coverage corresponds to the length of each gene. So we want to maximize the total length that our genes have. And if there's any ties, we want to use the score of the alignment to break ties. So that's how we infuse the signal from TRS from finding transcription regulatory sequences in how we use that to actually also find genes. So that's the problem we want to solve. So what does this correspond to? So let's take a look here. So the first step, if when given a genome sequence, actually let me talk one. I need to mention one other thing. So this V leader, V body, because our input is actually an unannotated genome. So how do we split an unannotated genome into V leader and V body? So what you can do is every coronavirus, the first gene that you'll find is 1A, ORF1A. This is a common feature to every coronavirus. And in addition, what you also have is 1A will have a slippery stop codon. And, there's going to be a frame shift of -1, and it will give you 1B. And so we know that that's kind of like the first, those are the first two genes that you need to see in a genome, in a coronavirus genome. And we also know that they span, about, between 50 to 75% of the total genome. It's a really long sequence. So those two genes are super long. So this is something that you can exploit. So you can literally search, for the first two ORFs, where the first ORF intersects with the second ORF, and there's a frame shift of -1, and together they span more than half of the genome. So this is something you can do very easily in polynomial time, and this will split your genome into two parts, a V leader and a V body. So that's essentially what's happening here. All right, let's talk about the method. So how do we solve this problem statement? So again, this is something that can be solved in polynomial time. And the reason why is as follows. So let's take a look here. So we have, we enumerate candidate open reading frames in V body. So you can do this very easily in polynomial time. So you get a set of candidate genes. Okay. And in addition, we're going to take the same strategy as the strategy that we used for solving the first problem. So we're going to slide a window of length omega through V leader. Okay. And essentially what we're doing is we're trying to find a TRS alignment for a subset of genes. So what you can do is you can actually solve so this, because we fix a window of length omega, this window U in V leader, we can solve a bunch of pairwise alignment problems between each candidate ORF and this subsequence that contains U in V leader. So that's going to give us a bunch of alignments. So that's what I'm showing you over here. So this is a dynamic programming table. You can see there's no gaps, so it's kind of like a diagonal line. So these are different solutions. And in addition, what we can do is we can encode the length of the corresponding gene, and also the score of the alignment of the pairwise alignment. We can encode that for every gene, and we can do that in the form of a graph. So each node in this graph is going to correspond to a candidate ORF, and the score or the weight of the node will be a combination of the node weight. And we use as a tiebreaker the score of the alignment. So there's actually a second factor here, a second term that I'm not showing. And what we're trying to do is we're trying to find, an independent set. So a subset of nodes that don't intersect, they don't have any edge in common, with maximum total weight. So this is a maximum weight independent set problem. So maximum weight independent set is an NP-hard problem for general graphs. But in our case, the graphs correspond to interval graphs because, there's an edge between two nodes if they intersect, if their intervals intersect. So we have an edge between x1 and x3 because they overlap over here. But there's no edge between x1 and x2, no edge between x1 and x4, etc. So this particular type of graph, a lot of, for interval graphs, a lot of NP-hard, graph problems actually become solvable in polynomial time, and independent set is one such problem. So you can actually solve this whole problem, this maximum weight independent set problem, in polynomial time. And this is the total running time that we have. So we solve this for each window U. So we keep sliding this window U. So that's why we have this V liter squared over here. And this is the total running time. Good. I'm just going to pause here. Any questions about this part?
Mohammed, I had a question, but I'm worried
Yes.
that you're going to cover it in the results section.
Go ahead.
Which is that the TRS alignment is used, you're saying, to break ties because there could be many independent sets of the interval graph with equal weights. And I was curious about how often you find ties getting broken.
That's a great question. Let me see. Let me get back to that. I don't think I have
Okay.
a particular result slide, but I do want to talk about. We could actually look at some results together because I've seen this several times. So we should be able to find cases where that happens. But you're right. So because I'm using it as a tiebreaker, so kind of like the main signal that I'm using here is genome coverage. And then, yeah, and then I'm kind of like using the TRS alignment score as a tiebreaker. Yeah. So the only way where the tiebreaker would be active is if you had multiple different sets of genes with the same total length. Yeah. So I'll talk about that in a minute. Yeah. Okay, let's take a look at results again. So there, to summarize, we've looked at two problems: TRS identification, that was question 1, and the TRS plus gene identification, that was question 2. So CORSID A, that was the first algorithm that I introduced, solves the TRS identification problem, and CORSID solves the joint inference problem, the TRS plus gene identification problem. And both, are polynomial time algorithms. So both problems are actually easy, which is kind of surprising. So multiple sequence alignment is really hard, but because we have this dependency because of the core sequence that they must have in common, it actually becomes solvable in polynomial time. So what we see here is the performance of CORSID in terms of finding TRS-L. So one thing that I haven't mentioned, so ground truth, so the actual location of TRS-L that we use as ground truth is based on secondary structure, plus also a multiple sequence alignment of the leader region. So we actually computed multiple sequence alignment, and then we also looked at secondary structure. So there was a lot of manual correlation here. And you can kind of like see there's a drop in performance from CORSID A to CORSID. And the reason why is because CORSID A is actually using the gene locations as input. And here in CORSID, we're trying to find the genes simultaneously. So you can see that, about 5% of instances, we don't identify TRS-L accurately. But this is still, a good, we still perform pretty well. So that's reassuring. And the other thing that we did is we also wanted to assess the performance of gene identification. So what we did is we took, so we have 468 different coronaviruses, and we downloaded all their annotations from GeneBank. So we downloaded a bunch of GTF files. So what we found is that several of these GTF files were actually incomplete. They did not span the genome. So what we ended up doing is we ended up blasting, doing a lot of blasting. So we blasted like all the genes that were actually present or annotated against every other sequence. And we kind of like improved the annotation. So we got a better idea of like, what are the genes that are actually really present. And so this was done using BLAST, and we used like super restrictive criteria in terms of like when we actually add a gene to the ground truth. But anyhow, so what I'm comparing here is the performance on gene identification. So there's precision and recall, because each method identifies, a set of genes. So you could ask yourself, which fraction of genes does it recall, and what's the precision? Like, does it identify genes that are actually not there? So ideally, you want precision and recall to be 1. So if precision and recall are 1, it means like you recovered exactly the set of ground truth genes and you didn't identify anything else, which is good. So what we can immediately see is I'm showing you here the results of all 468 instances and all gene. So there's actually 468 dots in each in each box plot. So Glimmer and Protocol are relatively old methods, and they're more used for metagenomics and bacterial genomes. VADR is more recent method, and it's specifically designed for viral sequences. So you can see that VADR actually performs better. And it's no surprise that there were actually 50 coronavirus genomes that VADR actually used in their training. So it's highly, geared towards like viral genomes, and in particular also coronaviruses. So what we see is we have roughly similar performance as VADR, but we outperform them in terms of recall. And that's actually also beautifully illustrated in this plot over here. So we're looking at recall and precision. And each dot in this plot corresponds to a specific coronavirus genome and the color indicates the method. So you can see that we achieve a really good trade-off between precision and recall. So that's actually also indicated with this dark green contour here, which is like close to the top right corner. So that's good. And the other thing that we built, so these are 3 undergrads. We built this web application to actually look at the solution space of different coronavirus genomes. So you can actually start answering like Aaron's question, and oftentimes, if you're an expert user of this tool, you typically want to actually look at the solution space because maybe the first solution is not quite right. So you want to be able to kind of like look at all other solutions and then select the solution that you deem to be more correct yourself. So that's what this web app is supporting. And in conclusion, so what we did here is we introduced CORSID, so the first method to simultaneously identify TRS sites and genes. And the cool thing is it enables de novo identification of TRS sites and genes. And the use case would be, you have a new coronavirus genome, you've never seen it before. So there's no annotation. You just have the assembled genome. And you can use this tool to get an idea of what are the genes present in the genome and also what are the TRS sites. And you can use the TRS sites to kind of like classify it, like what type of coronavirus is this? Is this an alpha, beta, gamma, or delta coronavirus? And you can even go down to the subgenus level. So it outperforms state-of-the-art methods. And in terms of future directions, there's a couple of interesting things. So we want to look at we want to support alternative start codons. So there are a couple of coronaviruses where genes don't necessarily start at ATG, but they start using a different start codon. So we want to, take that into account. And we also want to look at Kozak sequences. So these are motifs that the ribosome, detects and uses to kind of like initiate translation. In addition, a lot of genomes were actually incomplete. They were truncated, so they did not have TRS-L. And as you'll notice, there's a huge dependency on TRS-L because it's essentially a star alignment with, as the center star, we have the TRS-L sequence. So, this approach would break down if the input genome that you have, the input genome sequence, does not, misses this front part, this 5' end. And then the other direction that I would love to explore beyond coronaviruses. So if you look at Mesoniviridae, so actually let me just jump here. So this is another family within Nidovirales. So Mesoniviridae, these mainly affect, they occur in mosquitoes and also, mainly mosquitoes, but there's something interesting happening there. So instead of a single TRS sequence that recurs, that's present at the beginning of the genome in the leader and occurs in front of every gene, they actually have 2 different TRS sequences. And some downstream genes have both TRS sequences and sometimes only one. So there's a beautiful generalization of this problem to the case where you have like 2 different alignments. So we want to look at that. And so in terms of acknowledgements, so I want to acknowledge my graduate students, Choi and Palash. So Palash is now a postdoc at Princeton University. And I also want to acknowledge the 3 undergrads who built a beautiful web interface for this tool, funding agencies, and CORSID is available. It can be installed using Bioconda. All right. Thank you, everyone. So, Aaron, you had a question about finding or like how often does it happen that we actually use the tiebreaker. So let's take a look at that. So this is also a good opportunity to. Yes?
I just want to remind everyone that if you don't if your mic's not working, for example, you can put questions in the chat and I can read them to Mohammed. So as we're looking at data, also, you know, now is a great time to type your questions. So the other idea I had as we're looking through stuff was, I'm imagining the cases where an expert user might disagree are cases where you pick something where there's not a tie, but it's like marginally better in terms of coverage, and then you maybe really prefer something of
else,
lower
like
total weight because
happen.
of the TRS alignment. And so an expert user would say like, no, this other one is better, even though the total weight is slightly less or something.
Yeah.
So you almost have another formulation of the problem, but with like an error bound on like how close you have to be to the total weight to then engage like a tie-breaking mechanism or something, or some kind of
That's
other.
fair. That's a good point. Yeah. No, you're absolutely right. I think the beauty of this formulation, or like the beauty of the problem setup is, you know, you actually get access to the solution space. So that's what I'm showing you over here. So this is SARS-1, not SARS-2. And what you see here is like these solutions. So the top 4, they had the top 5 actually all had the same genome coverage.
Oh, wow.
And you can kind of like see that they actually have different TRS alignments, but they span the same genes. And this is the solution that was picked. So it was the top one because it had the best TRS alignment. So actually, if I pull up the second one, let's just add it. So, you know, it's just like a score of 1 less. So it's mostly identical, but where did the difference happen? Let's see. Yeah, there was a difference in S and also a difference. So it's just a slightly different trade-off. So S and E differ a little bit, but it's just a difference of alignment. But we're still spanning the same genes. So you can literally explore the solution space. And there are indeed instances where, you know, we did not find the right solution. So where, you know, we actually, I think that happened like 5% of the time. And then if you would like use this tool, you can typically like, you know, opt for something that has like slightly lower genome coverage, but a better TRS alignment. So there is indeed a trade-off. So it's a multi-objective optimization problem, and we use the TRS alignment as a tiebreaker. But you could actually think about, you know, thinking about like Pareto optimal solutions, like the space of Pareto optimal solutions between the 2 objectives. Yeah.
You could also think about, yeah, I mean, that's so cool. I think your point that the best part about this problem is that now you see the solution space, and yeah, it's really cool. And almost it's characterizing like uncertainty in it that the user can engage with.
Yes, absolutely. Yeah, that's really cool. And there are a couple of other interesting points that I should mention. So actually, so what I said at the beginning is not, so biology always has like a lot of exceptions. So SARS-CoV-2 is like pretty clean. So every downstream gene has like a unique TRS sequence, pretty much. 7B, not so much. So, but other coronaviruses, like you could have 2 different genes that share the same TRS. So what that actually means is, you know, the way this works biologically is there's something that's called ribosomal leaky scanning. So the ribosome won't necessarily start translating at the first ATG. It can actually skip over it with a certain probability and then move on to the second ATG. So that's how you can get 2 different genes using the same messenger RNA. So, and that's something that we don't incorporate in this. It's not part of the problem statements, but we could actually think about that. So because right now what I did was like this thing over here that each TRS alignment induces a gene set. And each TRS-B gives you 1 gene, but it might be the case there are actually 2 genes because of ribosomal leaky scanning. So that's another direction that, you know, should be explored.